Welcome to APSC webinar series supported by the Japanese Circuit Society, Cardiovascular Precision Medicine. I'm Issei Komuro, professor at the University of Tokyo, a vice president of the International University of Health and Welfare, and I'm also the immediate past president of APSC. APSC has been doing lots of webinars, and the title of today is Precision Medicine. Precision medicine, also known as personalized medicine, is an innovative approach to a medical treatment and healthcare that takes into account individual variabilities in genes, environment, and lifestyle for each person. Rather than using a one-size-fits-all approach, precision medicine aims to tailor medical care and treatments to the specific characteristics and needs of each patient. The key elements of precision medicine include personalized risk assessment, early detection and prevention, tailored drug therapies, and data integration. And to enable precision medicine, basic research, such as research on genomics, omics, and IPS, is really important. In this webinar, 4 leading scientists from Asia will talk about excellent basic research leading the precision medicine for cardiovascular diseases. Have fun while you study.
Thank you for the introduction from the Professor Komuro. First speaker is Dr. Kaoru Ito. Please see his talk.
First of all, I'm very sorry to present in the recording, so I'd like to share my slide. Today, I'd like to talk about the genomic analysis of atrial fibrillation, AF, and the perspective of precision medicine realized by the result. I have no COI to disclose. So clinical causes of AF are known to be lifestyle factors, other diseases affecting the heart, and non-modifiable factors such as age. On the other hand, what landscape would we see from the viewpoint of genetics? Actually, from genetic perspective, cardiovascular disease can be divided into 2 categories. The first is mosaic disorder, where a single genetic variant, usually very rare with a large effect, plays a critical role in the disease development. Ion channelopathies, familial cardiomyopathies, and familial hypercholesterolemia fall into this category. On the other hand, there is a polygenic disorder, caused by the accumulation of a large number of genetic variants with high frequency but a weak effect. This applies to common cardiovascular disease such as coronary artery disease and AF. From here, I'd like to focus on AF as a common cardiovascular disease. So, first, I'll talk about genome-wide association study of AF. For genomic analysis of common disease such as AF, genome-wide association study, GWAS, is very useful. Genetic variants are detected comprehensively across the entire genome, and case control study is performed using each variant as an experimental variable to identify loci associated with the disease. Multiple studies can be also combined for meta-analysis to increase statistical power. However, loci identified by GWAS are often not in a protein coding region, so we should take care of the interpretation. Which means it sometimes happens we are not sure which gene is affected by a variant detected by GWAS. Therefore, omics information from other layers is required for the interpretation of GWAS. Currently, functional interpretation is performed mainly using epigenetic information which can be easily mapped to DNA regions, and transcriptome information, which can also be dropped into gene expression, while other omics information with single-cell resolution can be also utilized. I'll skip the explanation this time. GWAS augmented with multi-omics data will not only elucidate disease biology, but also through polygenic risk score, causal inference by Mendelian randomization, genomic drug discovery, it will contribute to the realization of precision medicine for cardiovascular disease. Alright then, let's get back to AF. This slide shows the history of AF GWAS landmark papers. As you can see, the population included in these analyses were mainly Europeans, which means East Asians are underrepresented. Initially, just 550 cases were included in the study. In 2014, the paper of the AF-Gen Consortium, an international consortium for AF genetics, was published. Later, in 2018, when the UK Biobank started to be included, the number of analyzed individuals increased dramatically, and we published the latest AF-GWAS paper this year. This slide shows the result of the AF-Gen Consortium paper in 2018. We are part of the AF-Gen Consortium and try to clarify the AF mechanism from a genetic perspective. In this paper, we conducted GWAS with approximately 600,000 individuals, and we found that the mechanism of atrial fibrillation is not only related to genes involving electrophysical functions such as ion channel systems, which had already predicted, but also genes important for cardiomyocyte contraction and circumstructure, which are shared by cardiomyopathy, and to genes related to the heart development, which are shared by congenital heart disease. In short, through a genomic analysis, we have learned that AF is not just an arrhythmia, but a complex disease with several aspects similar to other cardiovascular diseases. Additionally, we have identified a new key molecule for AF in our paper published this year. As mentioned in a previous slide, just looking at DNA in GWAS alone is not enough to understand the disease biology. We integrated our analysis with other omics information. First, by integrating transcriptome information, we've identified IL-6R as a key gene for atrial fibrosis in AF, which had not been pointed out by previous GWAS. Second, by integration with epigenomic information, we identified estrogen-related receptor gamma as a key transition factor for AF development, besides TBX5. Through these analyses, we have succeeded in revealing new aspects of AF. Next, I'd like to move on to the topic of the potential clinical application. As you all know, we not only use GWAS to infer disease biology, but also use the result to estimate the genetic risk of a disease for each individual. In particular, genetic risk score developed using the weights of variants obtained from GWAS is called polygenic risk score, PRS. PRS then represents the cumulative genetic risk for disease development, which means individuals with higher PRS have higher genetic risk of a disease development and vice versa. However, as we expected from the population specificity of the genome, there is also population specificity in PRS, which means European PRS does not work well in East Asians. So, we developed high-performance AF PRS for Japanese using the result of Japanese and European GWAS. Here is a figure that shows the performance of AF PRS we developed for Japanese. Compared to the bottom 10% peers group, the top 10 peers group had an odds ratio of 4 or higher for AF development, regardless of family history, indicating the ability to distinguish AF cases from controls. PRS is a predictive score genetic risk for disease development, which have been validated several results where AFPRS predicted the AF risk before the onset. In this scenario, it may be possible to predict and prevent the disease onset in the early stage without the information of clinical risk factors. Such possibility was already suggested by several papers. On the other hand, the extent to which PRS can be applied depends on the disease-specific context. In AF, what are other applications? We explored it from the viewpoint of prognosis and complications. So, we examine the relationship between AFPRS and cardiovascular death from a prognostic perspective. The figure on the left shows the prognosis of patients who were divided into 3 groups: high PRS, low PRS, and the others. And we found that the high PRS group showed high cardiovascular mortality rate. This suggests that AFPRS may be also useful in estimating the prognosis. Since cardiovascular death includes a variety of cardiovascular diseases, the figure on the right breaks down the content. Where deaths from ischemic heart disease and heart failure were not significantly different between groups, while stroke death showed significant difference in the high PRS group, which is an important complication of AF. The result in the previous slide showed that AFPRS significantly stratified patients with stroke death. This could be a promising result that AFPRS may be used to predict a complication. Now then, in patients without AF diagnosis, can we predict cardioembolic stroke using AFPRS? We investigate this question using independent samples from Biobank Japan. Using approximately 100 to 20,000 Japanese without AF diagnosis, we examine the association of AFPRS with stroke phenotypes. The results showed that AFPRS was significantly associated with cardioembolic stroke, while we found no association of AFPRS with other atherosclerotic infarction and hemorrhage. This result suggests that AFPRS predicts the risk of cardiogenic stroke in patients without AF diagnosis. In summary, our results suggest that AFPRS may be one of the effective tools for early detection of the disease, prediction of disease prognosis, and the prediction of cerebral embolism. In addition, the genomic information used in the AFPRS can be obtained regardless of age, inexpensively, and once the score is calculated, the value could be used forever, especially because it's important for AF patients to prevent stroke before it occurs. AFPRS may be used as an indicator for the introduction of anticoagulants or left atrial appendage closure devices, if possible clinical evidence accumulates. Such a possibility was suggested. This is the end of my presentation. Finally, I'd like to thank all the collaborators who helped us a lot in carrying out this research. Thank you very much for your attention.
Thank you, Dr. Ito. We would like to discuss the presentation from Dr. Ito. Are there any questions or comments?
I have first question, Seitaro. I wonder what you think about the finding of the 2 new candidate genes that was pointed out in the Nature Genetics publication. Congratulations first, by the way. So one of them is an interleukin receptor and the other estrogen related gene. This implies that inflammation has much to do with atrial fibrillation. Maybe it's through other structural reasons of the myocardium, etc. But we are currently not using inflammatory approach to treat atrial fibrillation. I wonder whether on clinical side people are thinking about this.
Yeah. Your question is about the interaction between the development of atrial fibrillation and the inflammation.
Mm.
In the genomic analysis, we found that the IL-6R is one of the important candidate gene related with the development of atrial fibrillation. Yeah, we think that IL-6 signaling may be important to cause atrial fibrillation. So, we have
Did you do, sorry, interrupting. Did you do something like you regressed out cardiomyopathy?
Yeah, no, we have not yet. Yeah.
Whether the cause of inflammation is direct to atrial fibrillation or via structural heart disease, disease.
Actually, we have performed the functional analysis regarding the related with the mechanism with the ERR gamma. Yeah, actually, we used the inhibitor of ERR gamma to iPS-derived cardiomyocytes to show that this inhibition actually leads to the dysfunction of the normal rhythmic cardiomyocyte contraction and abnormal gene expression profiles. But we have not performed the experiments regarding inflammation. Sorry.
Very nice. Congratulations.
Can I answer the question?
Thank you.
As you may remember that more than several years ago, there was a CANTOS trial which used anti-interleukin-1 beta antibody for the patients with high CRP. And the results is very remarkable. Anti-IL-1 beta antibody significantly reduced cardiovascular diseases and also some cancer. And now the anti-IL-6 receptor antibody clinical trial is going. Probably a couple of years later, we'll get some results. So we'll see. Thank you very much for asking.
Yeah, I may have a question as well. So very happy to hear the excellent study. Congratulations. And from the polygenic risk score and imputing some of the causal genes, do you come across genes that are not specifically expressed by cardiomyocytes? And yeah, the context of this question is to find out other non-cardiomyocyte mechanisms. Yeah.
Yeah, we have analyzed the expression profiles using single cell data. Actually, many genes are expressed by from cardiomyocytes, but the other many genes are also expressed from the other cell types, including endothelial cells or fibroblasts and so on. So, yeah, we would like to know the function of these other cell types than cardiomyocytes. Thank you so much.
Thank you.
Actually, our colleague, Dr. Yamaguchi, has get more than 500 biopsy samples of atrium. Yeah, so his first paper has been published in the European Heart Journal recently. So please look at it.
Seitaro, I have one more for clinical angle. So the atrial fibrillation analysis, did it also include patients with paroxysmal atrial fibrillation? So in atrial fibrillation that is intermittent, because sometimes we have patients with stroke, and it was really nice that you did the analysis with cardioembolic stroke, because these are the patients that sometimes you don't have the cause for the stroke, and it may be they have paroxysmal atrial fibrillation. I wonder whether this PRS is picking those people up also.
Yeah, we included paroxysmal and and persistent. So yeah, we think that by using the PRS of atrial fibrillation, we can identify this type of atrial fibrillation.
That's very nice. It's very powerful. Congratulations again. Very, very nice.
Thank you so much. Okay, so the next presentation is from Dr. Christine Cheung, and the title of her talk is Autoimmune Basis of Endothelial Dysfunction. Please go ahead.
Thank you, Seitaro. Yeah, hi, my name is Christine Cheung. I'm an assistant professor at the Nanyang Technological University. I am a vascular biologist. So I run the laboratory of molecular and vascular medicine. And as a step towards more personalized medicine, for the past years, our lab invested efforts to recreate patients' own blood vessel models. So one of it is through human-induced pluripotent stem cells, where we derive endothelial subtypes, mapping arterial venous axis, also trying to advance organotypic endothelial differentiation protocols. And also there's vascular smooth muscle cells where we can try to build and construct together endothelial under even dynamic culture system to provide a more physiological context for the study of these patients' model. And also, this has been extended to derive blood outgrowth endothelial cells directly from patients, where we believe that some of this primary endothelial cell culture can really recapitulate the molecular pathogenesis and some of the phenotype disease hallmarks in the cell culture. So here is some examples of our recent findings where we try to deconvolute in a few studies the endothelial disease biology. We also apply single cells transcriptomics to work out the molecular mediators. And then design a fit for purpose phenotypic assays so that we can understand endothelial pathological behaviors in a way to address the pathology that are manifested in the patients, from inflammation to pathological angiogenesis to some of the atherosclerosis hallmarks, where we are looking closer to endothelial immune crosstalk. So that actually leads on to some of our ongoing dysfunction. So in fact, autoimmune mechanisms remain rather undiagnosed in patients with low-grade chronic inflammation. Yeah, so actually a lot of conditions and comorbidities with metabolic dysfunctions, okay, and those patients will known to have higher susceptibility to adverse cardiovascular outcomes, and chronic inflammation is now recognized as underpinning etiology as well. So I think it's well known that some of the trials have been also been very successful to control and delay the first primary cardiovascular events. But it's also known that in these patients, despite gold standard treatment to control for cholesterolemia or glycemic endpoints, there's still a subset of patients with residual inflammatory risk, and it's characterized here by a level of CRP. And so there are still questions about are there other inflammatory mechanisms or risk factors that we have not accounted for. So pertaining to that, I think we know that a lot of us are COVID survivors, and the long-term cardiovascular outcomes are being reported. And there have been also concerns about, are we really shifting the graph whereby accelerating the first primary cardiovascular events for people post-COVID. So during acute infections, there have been some studies to look into autoantibodies and whether they are raised against certain tissue-associated antigen. And in fact, in this particular study, we were quite intrigued that they found and detected functional autoantibody against the immune system, vascular system, as well as some of the chemokine and cytokines. But there is still questions about, are these hallmarks persistent post-COVID or generally in patients with chronic inflammation? And as we start to understand from the field of autoimmune disorders, actually we learn a lot from there. It's very well established that the mode of action of the autoantibodies can be targeting against autoantigen on certain tissues, and that can activate a complement system. Yeah, with the formation of the membrane attack complex that then creates tissue damage, but also autoimmunity in terms of the interaction with some of the cytotoxic T cells or NK cells. And that wealth of knowledge is still being studied or characterized mainly in patients with autoimmune disorders, or even HIV patient that's known to have chronic inflammation, but it's not a routine as part of the care management of disease management for a lot of patients who have the other cardiovascular risk factors. So this leads to some of our more recent research questions about will endothelial cells raise some of these autoantigens and be induced under chronic inflammation, and whether there are endothelial targeting autoantibodies that can contribute to vascular damage, and what are the mechanisms of this autoantibody-mediated dysfunctions. So we got quite intrigued when we look at this autoantibody profiling that was done during the acute infection phase of COVID. So now, in collaboration with the National Center for Infectious Diseases, Singapore, they have a cohort of post-COVID survivor, and that we can understand in these, based on cytokine profile, whether they sustain persistent inflammation based on cytokine hallmarks, but also for us to start to look at do they have persistent autoantibodies that have reactivities against vascular tissue of our interest. So we first did data mining to understand the expression or genotypic expression of some of the counter antigen of those autoantibody, and this is the data set that we try to do data mining. So from that organotypic endothelial data set in mouse, we look into a few of those potential antigen that are highly enriched in blood vessel endothelium. And also here, just to do understanding of the subtype of endothelial cells that this may be more relevant. So generally, we are seeing more on the venous endothelial cells that have some of these key antigen. But of course, the good news is baseline individual, we are not expecting, or you shouldn't be expecting actually autoantibodies that is really rendering immunogenic attack on the blood vessel. But what happened in inflammation is that when we subject primary endothelial cell culture to plasma samples of COVID recoverers, okay, those who recover from mild versus patients who recovered from severe COVID who needs oxygen supplementation, we see upregulation of some of this antigen under the plasma stimulation. And then we went on to custom make a protein array to screen for some of this what we believe are anti-endothelial autoantibodies. So vast majority of patients, okay, do have very baseline or if not negligible level of autoantibody. So I think that's still good news. But this never linear. That means we are able to detect sporadically some patient that may sustain a higher level of these anti-endothelial autoantibodies. Yeah, so I think the pie chart here will demonstrate that patient that maybe is post-acute infection, we can pick up a few of such individual. Yeah, but the vast majority, I think, is at a rather negligible level of this. So, of course, that got our interest to understand what it means to have the presence of these anti-endothelial autoantibody in these patients. So, to understand the impact, we use a cellular biomarker that is associated with vascular injury, and that is to characterize the circulating damaged endothelial cell in these patients. This is a rare population that can be found in the bloodstream. It is it represents the entity of dislodged endothelium from the site of vascular injury. So they are present in the bloodstream. And it's we have been able to characterize the level of circulating endothelial cell across various types of patient, from those with coronary artery disease to metabolic dysfunctions, and now with COVID survivor as well. And it's also demonstrated in other prior studies about its relevance to understand in situ vascular injury. So we try to correlate here. We do achieve a rather modest but significantly positive correlation of the anti-endothelial autoantibodies with the level of these circulating endothelial cells. Moving on is that to better understand the mode of action of these autoantibody is that we try to block this autoantibody with blocking peptide or the recombinant protein that represent the antigen. The design rationale for blocking peptide usually is to synthesize the peptide that represent the extracellular binding domain of some of these antigen. Yeah, so I think this is the outcome from trying to screen different synthesized peptide to find the one that is actually the binding domain between the cell antigen with the autoantibody. And here we again did plasma exposure to primary endothelial cell culture, and also did this so-called intervention in vitro with blocking peptide and recombinant protein, and that this is if some of this were effective in averting or reducing the level of human immunoglobulin binding to the cells. And that with the blocking peptide recombinant protein, we also show that it can significantly reduce the level of apoptosis, endothelial apoptosis, when these cells are subject to the COVID patient's plasma. Yeah. So to round this up, I think moving forward, we do aspire to more comprehensively construct molecular atlas of these endothelial cell surface autoantigen based on patient-derived endothelial cells as a more personalized models. And that the outcome of this, we hope that we can lead to more precise treatment strategy, especially when it comes to specificity of autoantibody, even neutralizing these autoantibodies and minimizing immunological attacks on the blood vessels. Yeah. So with that, this is the work of actually Issei is my postdoc who's driving a lot of this part of the work, and also our huge support from the collaborative network in terms of samples and patient profiling. Yeah. So thank you very much, and welcome your questions.
And thank you, Christine. We would like to discuss her presentation.
Can I ask questions?
Yes.
Christine, excellent studies. Congratulations. I'm very interested in your results. But I have a couple of questions for you. So first, you mentioned that COVID-19 survivor plasma upregulates endocellular specific antigens.
Mm-hmm.
So what molecules induces upregulation of the endocellular antigens in the plasma?
Yeah, very good questions. So we believe that it could be the cytokines and chemokines that are in this COVID plasma. So with that, which we didn't show that, so we did a Luminex cytokine profiling on the COVID plasma. So from there is to identify patients that have persistent inflammation. Yeah.
Mm-hmm.
So some of this will cover CRP, interleukin-6, IP-10, and typically we've found that a huge number of such cytokine and chemokines, they correlate directly with the circulating damaged endothelial cell level. So firstly, we think that endothelial activation is definitely a direct consequence of the persistent inflammation that is featured in some of the patients. So I would like to spread the good news is actually 1 to 2 years post-COVID, actually a lot of these inflammatory hallmarks, they do return to baseline. They do return, but we are probably picking up those who may still sustain a chronic or residual inflammatory risk and how that still impact on endothelial dysfunction. Thank you.
Second question is, what is the definition of circulating damaged endothelial cells? As you know, more than 20 years ago, endothelial progenitor cells attracted much attention, but the definition is still not clear. So I wonder, what is the definition of your damaged circulating endothelial cells?
Yeah, that's a great question. In fact, I recently wrote a review to talk about the difference of blood endothelial cells. So I believe what you have mentioned about is endothelial progenitor cell, the EPC, that's most well known. It has attracted the most attention because of its potential for regenerative medicine, and that it represents a postnatal pool of adult stem or progenitor cells that are often believed to home to the sites of vascular injury to actually induce neovascularization or directly take part in that. So EPC, the traditional view of endothelial progenitor cells, are those that are mobilized from bone marrow. And recently, I think in the vascular biology field, it could also represent maybe some of the vascular resident pool of stem cells. Yeah, so I think, so there's, and people have come up with some of the more technical oriented nomenclature, that they no longer only define APC based on markers because they have an overlap on marker, some of the immune cell subsets, but rather is function in terms of inducing angiogenesis. The biomarker that we are using is very different. It's the circulating endothelial cells. It has a immunophenotypic profile of mature endothelial cells. So the way we discern from the endothelial progenitor is they actually have mature endothelial cell marker. And secondly, the origin of these circulating endothelial cells come from damaged blood vessels. So they don't have any regenerative capacity. The circulating endothelial cell, even when we culture them, there's no way we get them to further maturate or do any more sprouting angiogenesis. So they represent a pool of damaged mature endothelial cell in the bloodstream where we use it as a biomarker for vascular injury. So in fact, if we look at, people have been characterizing more on the class of endothelial progenitor cell, and actually the more means maybe they have better prognosis instead. But in this case, it's the reverse for the circulating damaged endothelial cell, where the more we are seeing in terms of the level, there is a surrogate marker for vascular damage. Yeah.
Okay. So last short question is, so you seem to develop endothelial cells and the smooth muscle cells from iPS cells, right?
Yes.
We developed the cardiomyocyte from iPS cells, but iPS-derived cardiomyocytes so immature, so they don't have T tubules, they don't express high levels of the cardiac genes. So how about endothelial cells and smooth muscle cells derived from iPS cells?
Yeah, it's a great question. I think all the vascular differentiation, we will do functional characterization. So the definition of really functional endothelial cell is lumen formation. So the gold standard testing, at least is if it's post-transplanted in mouse, we want to see there is real perfusion and integration into host vasculature. So that was possible, at least from our end, the endothelial cells and small muscle cells that we made, we have seen in vivo integration and the perfusion of blood. Yeah. So in vitro wise, so I think it's all the phenotypic assays that we have done, looking at tube formation, 3D sprouting angiogenesis. So functional attributes, I think we are recapitulating to a certain degree of success. But if we look at really at the molecular hallmark, and the clustering compared to real mature primary cells and fetal endothelial cells, I think the general whole transcriptome may still suggest that they are more on the more closer to fetal endothelial cells. Yeah, but there are more experimental strategies whereby we can grow these cells in dynamic culture conditions. So giving it the hemodynamic shear stresses, physiological shear stresses, and that has been known to maturate some of these cells further.
Yes, thank you very much.
Thank you.
Thank you. I have one short question. Endothelial dysfunction has been known to be related with not only the COVID, but also heart failure with preserved ejection fraction, HFpEF. Can the presence of these antibodies stratify patients not only with COVID, but also with HFpEF?
Okay. Very good question. So, because I think I speak from in the capacity of a scientist, is that we believe that what we are studying in terms of endothelial dysfunction and the group of endothelial autoantibody, it really transcends disease conditions. And
Yeah.
that if the disease condition have inflammatory etiology, be it post-infection or people who are susceptible to have heart failure, some of these vascular changes are known to precede a lot of the organ really severe damages. Yeah. So the key even when we talk about it, we use post-COVID as more of a reference cohort only, that we can do this and apply this to other patients as well. Yeah, and that vascular changes often occur earlier. Yeah.
Okay, thank you.
Thank you.
Inflammatory heart failure is also a target of, I think. Thank you so much.
Thank you.
Okay. So thank you, Christian. That presentation is from me. Hi, I'm Seitaro Nomura from the University of Tokyo. The title of my talk is Precision Medicine for Heart Failure. Pathological stress, such as hypertension, myocardial infarction, cardiomyopathy, valvular disease, and inflammation to the heart induces cardiac hypertrophy and subsequent heart failure. During these processes, each cell in the heart responds to these pathological stresses to show several gene expression profiles. So we have developed a single cell RNA sequencing analysis pipeline to dissect the stress response, cell trajectories, and cell-cell interaction during these processes. First, we apply this technique to heart failure model mice. Transverse aortic constriction to the heart induces cardiac hypertrophy and subsequent heart failure. We isolated cardiomyocytes and used them for single-cell RNA sequencing. Also, we performed single-cell RNA sequencing of non-cardiomyocytes. And these are single-cell RNA-seq profiles, which can identify cell types. By using these single-cell data, we generated ligand and receptor co-expression network in the heart, and we also generated ligand and receptor communication network. By analyzing these networks, we found that fibroblasts actually interact with many cell types, including cardiomyocytes. By focusing cardiac fibroblast network, we found that HTR3, which is a serine protease, is located at the center of cardiac fibroblast network. Actually, the expression of this gene is specifically expressed in cardiac fibroblasts. First, we generated HTR3 knockout mice. These knockout mice show cardiac hypertrophy, and mild pressure overload induces heart failure, cardiac dysfunction, and cardiac fibroblasts. So, and we also confirmed that downregulated gene profiles in cardiac fibroblasts in not only in mice and humans. We also performed mechanical stretch experiments and found that these mechanical stretch to the cardiac fibroblasts induced the downregulation of the gene expression of these genes. Next, we performed single-cell RNA sequencing of cardiac fibroblasts from wild-type sham, wild-type tag, knockout sham, and knockout tag. This is the single-cell RNA-seq UMAP, and we identified the two trajectories from the original state. And we found that the expression of extracellular matrix modules were specifically activated at these cells. Actually, these cells were mainly from knockout and with tag. These genes were specific for genes related with extracellular matrix, collagen fibril, and TGF-beta related signals. Therefore, we hypothesized that HTL3 may be related with TGF-beta signaling. Actually, biochemical experiments show that HTL3 directly interact with TGF-beta, and overexpression of this gene in cardiac fibroblasts leads to the extreme downregulation of TGF-beta protein expression level. So we conclude that the TGF-beta HTL3 binds to TGF-beta for degradation. And we also performed single cardiomyocyte RNA sequencing and reviewed that two trajectory from the original state. And we identified two distinct gene expression profiles. One is module 1. These gene modules were related with mitochondria and DNA damage repair genes. These genes were critically downregulated in cardiomyocytes after pressure overload. But whereas module 2 genes, which is related with TGF-beta and the p53 signaling, were specifically activated in the trajectory 2, only trajectory 2, which is mainly from knockout with tag. So HL3 knockout induced in cardiac fibroblasts leads to the upregulation of TGF-beta and the p53 signaling in cardiomyocytes. We also confirmed these expression profiles in humans. We isolated cardiomyocytes from patients with heart failure and used them for single-cell RNA sequencing. We also reviewed the trajectory from the original state. And the very similar expression dynamics could be confirmed. First, mitochondria-related genes were critically downregulated in patients with heart failure. And also, in some cardiomyocytes, we found that in some cardiomyocytes, these genes were critically upregulated. These module 1 genes include interferon, TGF-beta, and DNA damage responses. So, and we also found that several cytokines were expressed in these cardiomyocytes. So we used plasma proteome data from human heart failure patients to stratify, to identify the several cytokines or proteins to stratify patients with mild heart failure and severe heart failure. Random forest machine learning algorithm could identify that IGFBP7 was the most important factor to stratify patients with mild and severe heart failure. Actually, IGBP7 is show the good performance to stratify patients with advanced heart failure. So the summary of the first talk is here. HCl3 is expressed in cardiac fibroblasts, and mechanical stretch to the cardiac fibroblasts leads to the downregulation of these genes. And knockout or downregulation of these genes leads to the activation of TGF beta. This overactivation of TGF beta not only activates cardiac fibroblasts, but also induces DNA damage and p53 signaling and secretory phenotype in cardiomyocytes. So we also recognize that DNA damage in cardiomyocytes were important to stratify patients with heart failure. So we developed the molecular pathology analysis pipeline using endomyocardial biopsy samples by using antibodies against poly-ADP ribose. We could detect DNA damage positive cardiomyocytes like this. By using these mechanism, we can stratify DNA damage positive patients and DNA damage negative patients. Actually, the prognosis is very, very distinct. So, quantification of DNA damage predict left ventricular reverse remodeling in heart failure. Final talk is about genomics in heart failure. We have performed genomic analysis of dilated cardiomyopathy and show that patients with TTN TRK invariants show good prognosis, whereas patients with LMNA mutations show poor prognosis. So we generated LMNA mutation knock-in mice using CRISPR technology. These LMNA mutated mice showed heart failure and sarcomere abnormal orientation. In order to identify the molecular mechanism in the heart, we performed single-cell RNA sequencing and attack sequencing. First, RNA sequencing can stratify cell types, and actually, in contrast with wild-type mice, LMNA mutated mice have fewer mature cardiomyocytes. And we also performed the single-cell RNA sequencing and integration, and performed integration with RNA-seq data. This integrated analysis not only confirmed the downregulated population of maturated cardiomyocytes, but also these mature these mature cardiomyocytes were regulated by TED1 transcription factors. We actually confirmed that mutated LMNA is specifically binds to TED1, and immunocytochemistry show that TED1 transcription factors show that this location of this protein localization in the LMNA mutated cardiomyocytes, TED1 is observed at the nuclear membrane with the mutated LMNA. So we also finally confirmed that single cardiomyocyte expression in human heart failure patients, specifically cardiomyocytes from LMNA from patients with LMNA mutations showed very significantly downregulated expression of TED1 target genes. Actually, a chemical substance TT10, which activates TED1 signaling, rescue the transcriptional profiles in human iPSC-derived cardiomyocytes. And then we have developed the novel pathological model nuclear membrane trapping of TED1 by mutated LMNA perturbs cardiomyocyte maturation. So this is a summary slide. In heart failure, in this talk, we have showed the interaction between cardiomyocytes and the fibroblasts, and we showed the importance of the pathological significance of TGF-beta signaling. And we also showed them relationship between nuclear lamina and TED1 signaling. Yeah, thank you for kind interaction.
Seitaro, that's really so interesting. There's so many different pieces of information. I feel like I have so many questions to ask you. Can I start maybe with one, which is to ask whether the HTAR3 locus, does that have genetic signals from GWAS study before?
Yeah. Yeah. We have assessed the GWAS signaling, but there is no signal. But I think HTAR3 is the most interesting point is that HTAR3 expression is specifically expressed in cardiac fibroblasts. Yeah, but yeah, we don't know whether the genetic dysregulation of this gene may be related with the development of heart failure. But yeah, we have confirmed that actually downregulated expression of this gene in heart failure state.
Yeah, I mean, but whether or not there are human genetic SNPs in the loci of this gene, right? That also, for example, with EQTL on fibroblasts, and then from there, whether it appeared in heart failure GWAS or other atrial fibrillation GWAS, etc.
Yeah, great. And we have now conducted the integrated analysis using whole genome sequencing and transcriptomic and epigenomic profiles to identify the relationship between these genes with the transcription and epigenomic profiles. So yeah, we would like to answer this.
Very nice. Then maybe one more is quick one. Last one is I noticed that there are two types of the progression for cardiomyocyte mitochondrial DNA damage. Is it the mitochondrial gene expression? What do you think is explaining the two different directions?
Yeah, a great question, and we have reviewed the two trajectories, not only in mice and humans, and the most important point we found that the branch point during the trajectories were mainly activated in the p53 signaling. So we confirmed that p53 signaling activation at the branch point, So, but yeah, we did not understand the molecular mechanism during the diversification between the two trajectories, and that is it answer your question?
Yeah, may I ask a question too? Yeah, so that was really excellent talk. Yeah, really enjoyed it. So inundated with all the findings. So actually leading on to Roger's questions about the HTRA, is it known that your HTRA3 crosstalk with the other serine proteases? Because HTRA1 risk variance is well known associated with small vessel disease in the brain, also in the eye in terms of the macular degeneration. So I wonder, I mean, you found that was enriched in cardiac fibroblast. Yeah. So I wonder whether it actually should crosstalk with other serine proteases from the same class.
Yeah, it's also a very interesting and good question. Yeah, as you said, HTRA1 is very associated with the brain and brain disorder and so on. But actually, in the heart, HTRA1 is not expressed. So we would like to understand the molecular relationship between HTRA1 and HTRA3, but actually I cannot answer your question correctly, but I think several mechanisms may regulate the specific expression profiles of this gene in the heart.
Thank you. Yeah. I think there's a lot to learn there. Yeah.
Thank you so much. So, okay. So let's move on to the last speaker. It's okay. Last speaker is Professor Roger Fu.
Hello, Seitaro. Thank you for inviting me to speak. I am very glad to speak to the audience in the precision medicine seminar here, webinar, and also really good that both Seitaro, Christine, and Kaoru has laid the ground really nicely, especially for Kaoru's slides, where he showed that in precision medicine, we are also looking for targets in our gene discovery process. So this is one of the things that the Fu Lab has been working quite hard to do. Our mission is to work around cardiac epigenetics, gene expression control, and cardiac cell states, all in the effort to look for biomarkers and target discovery in feeding to the pipeline for precision medicine. So what we would like to be able to say is that in the years of working in Singapore, the lab has been contributing to many different discovered genes that we believe plays role in various pathways of heart disease, encompassing cardiac hypertrophy, cardiac cell states, cardiomyopathy, and coronary artery disease. Some of them are validated at cell level using iPS cell system, which Seitaro mentioned, also others using disease models, including the mouse. So I will present a project which deals with cardiomyocyte differentiation or development, where one of the graduate students in our lab has used CRISPR genome-wide screening to look for precisely candidates in the differentiation process of cardiomyocytes. And the PhD student is called Mick. And what Mick did was to make use of our platform of stem cell differentiation, which takes a very clear and predictable trajectory towards cardiomyocyte differentiation through the process of moving through to mesoendoderm transition, cardiac mesoderm, cardiac progenitors, and then to cardiomyocytes. And in each of those cell states, there are very clear markers that are used by the whole of the community to convince yourself that these cell states are transitioning. For example, OCT4 and SOX2 in the early state, and MYH6 myosin chain gene or NKX2.5 as the development of cardiomyocytes appears. So on the background of this platform, Mick applied the Brunello library, which has a set of guide RNAs, 77,411, each of the which targets each of the 20,000 protein coding genes, 4 guide RNAs per gene, applied to a pool of stem cells that are then put through the process of cardiomyocyte differentiation. So the aim with Mick's screening platform to carry this out was to look for necessary factors that are essential and when knocked out would then inhibit the development of cardiomyocytes. So upon his analysis, he found very consistent candidates that appeared in his screen, all to the right side of this curve of the black middle, indicating that these were the genes that were necessary and already quite well known for cardiomyocyte differentiation. Equally, there were negative controls, and these are the neuronal markers that acted as negative controls for us, where these were neutral factors towards cardiomyocyte differentiation. One of the genes that stood out in his analysis was a gene called NF2, which has never been implicated before for cardiomyocyte differentiation. And this was the gene that is well known to associate or causally linked to the disease called neurofibromatosis. And specifically, it's neurofibromatosis type 2. These are patients that develop benign neuronal tumors, and it can affect hearing loss as well as balance problems. But essentially, it is a tumor formation when you have loss of function mutation in NF2, benign tumor formation. And interestingly, in the literature, we observed and analyzed many publications and found that there were no reports of cardiac involvement in patients with neurofibromatosis type 2. So we asked ourselves the question why this was coming out in our screen when we are looking at cardiomyocyte differentiation. And I think we have the answer, which I'm happy to discuss further. So the first thing that Mick then went on to do was to create 2 clones of iPS cell lines of NF2 knockout. And he did that by using 2 sets of guide RNA. So the first set of guide RNA 1 was to knock out the gene NF2 by targeting a location upstream in the upstream portion of the gene, and the other with another set of guide RNAs to target the second section further downstream on the gene. The first knockout created an insertion of 1 base pair in a homozygote manner. So this was a complete knockout, homozygote knockout. And in the second set of clones, he knocked out 14 base pair deletion and a 1 base pair deletion in 2 opposite alleles. So again, creating a full knockout of NF2. And the first set of experiments he did was to carry out trilineage differentiation, and he showed that the formation of trophoectoderm, endoderm, ectoderm were not affected by the loss of NF2. And in fact, it is the formation of endoderm and mesoderm that was significantly perturbed. And also using the markers, as we mentioned in the first slide, for mesoderm and endoderm, Becurie, Fox A2, he showed himself and he convinced himself that NF2 deficiency led to the loss of mesoendoderm formation. In the full extent of cardiomyocyte differentiation, taking it through the protocol that I showed in the very first slide too, he was able to show that cardiomyocytes were not formed in the presence of NF2 knockout. The green here being a reporter that was being driven by myosin heavy chain. So the absence of the green showing that cardiomyocytes were unable to form. And not just cardiomyocytes that are unable to form without the NKX 2.5 marker, but actually also not being able in the first place to get to mesoderm with the loss of the UMS marker as well as the loss of cardiac progenitor. So the cells seem unable to even cross the transition of mesoendoderm to begin with. So at this point, we were quite convinced that NF2 deficiency led to the loss of cardiomyocyte formation. So digging deeper into the pathways of what NF2 might be, how NF2 might be performing its function at the molecular mechanistic level, there is some literature to show that NF2 has relationship to YAP signaling. So this is when we got quite excited because YAP signaling is quite a big portion of understanding of cardiomyocyte differentiation, cardiomyocyte biology. We found that NF2 knockout in the mesoendoderm state of day one differentiation, phosphorylation of YAP was completely abrogated or significantly lost in the presence of NF2 knockout. And one of the things that are really well known, canonical pathway of YAP phosphorylation is its upstream kinases, including MST1,2 and LATS1,2. And when Mick plotted for LATS and MST phosphorylation, he found no difference in the phosphorylation of these canonical pathway kinases, which then led us to wonder and scratch our heads. It doesn't look like the phosphorylation of YAP that was missing as a result of NF2 loss did not depend upon LATS or MST phosphorylation. And yet, it seemed that this would be still quite important for YAP because when Mick looked closely at YAP cellular localization, it was quite clear that in the mesoendoderm transition, YAP was in the cytoplasm, so it had localization in the cytoplasm, whereas in the knockout cells, the transition to the cytoplasm was not taking place, so YAP was instead in the nucleus. And YAP being a new nucleus, is also well known to be associated with the loss of phosphorylation on that serine residue of YAP. So this led to the conclusion at this point that we should pay attention to YAP nuclear relocalization as a result of loss of NF2. And what was then upstream of causing the change in phosphorylation of YAP and the nuclear mislocalization of YAP, we then discovered that NF2 actually has relationship to cell adherence junction in previous literature. And one of the key proteins in the cell adherence junction was a protein called MOT. And MOT phosphorylation seems to be significantly perturbed in the absence of NF2 as well. So this is when Mick decided to really dig deeper into the MOT, YAP, and NF2 relationship as results of the molecular interactions. In the meantime, we of course studied the RNA sequencing in great detail and showed very clearly that NF2 knockout cells are taking a very opposite trajectory, in red color here, compared to wild-type cells that undergo differentiation, transitioning from from pluripotent stem cells to mesoendoderm, to cardiac mesoderm, to progenitor, and then to cardiomyocytes, taking a really beautiful trajectory that is blue in color based on the RNA sequencing. When you have a loss of NF2, the trajectory is completely misdirected. And what we think results is epithelial-like non-myocytes instead of actual cardiomyocytes. So we proposed the model where NF2 is necessary to remove these roadblocks in order for the stem cells to carry on its transition down through the different cell states towards cardiomyocyte. So the summary box here saying NF2 necessary to remove the roadblocks en route to cardiomyocytes. So going back to the molecular mechanisms, because of the lack of MOT phosphorylation, Mick then went on to dig deeper to see if these three proteins indeed exist in a protein complex together. And he did a series of immunoprecipitations, pulling down, in this case, YAP, and blotting for NF2 and showing that it exists, and also doing immunofluorescence to show the presence of these proteins at the cell adherence junction or the cell membrane. Equally, he has done proximity ligation assays to show the three protein interactions with each other. And the loss of NF2 results in YAP falling off this protein complex. So what we believe we have been outlining at this moment is the formation of a complex at the cell adherence junction that was very necessary at the day 2 stage of mesoendoderm transition. So at this stage, we then went even deeper to the protein of NF2 and discovered that NF2, because it's so well known in the study of those benign tumors of neurofibromatosis, it is known that NF2, which has a firm domain, is a protein that does have interactions at the cell adherence junction, as I mentioned, and a lot of proteins that are at the cell membrane, under the cell membrane, do carry firm domains. Firm domains are characteristic of cell adherence proteins. And firm domain, an NF2 is a protein that exists in both a closed state and open state. And either of these closed state and open states are significantly impacted upon by the phosphorylation of serine residue 518. And moreover, the open and closed states are conferring its function for growth tumor suppression and is very much related to the function of NF2 in the cause of neurofibromatosis and benign tumors. What's also interesting when we read further deeper into the firm domain was there is a blue box motif that is sitting in the firm domain that comprises of 7 amino acids. So what we then did was took all of this information and decided to reintroduce back wild-type NF2 and various mutant forms of NF2 into the background of the knockout cell of NF2 to demonstrate the rescue of cardiomyocyte differentiation. So on the background of a knockout cell, we use blue fluorescent proteins as control with the flag tag, a wild-type version of NF2, open reading frame, a mutant form of NF2 where the serine residue was mutated to alanine so that it's non-phosphorylatable. We also mutated serine residue to aspartate to make it like a phosphomimetic, and then deleted the blue box motif, the 7 amino acids, and also created alanine mutation to all 7 amino acids. So several forms of mutant NF2 plus the wild-type NF2. And when he reintroduced each one of these constructs back into the knockout cardiomyocyte, he was able to show that the rescue was possible using wild-type NF2, of course, and also with the phosphomutants NF2. So phosphomutants did not seem to play a role in determining cardiomyocyte differentiation, whereas it was the blue box domain mutants that were still unable to rescue cardiomyocyte formation. And at the same time, he was able to show the nuclear localization was coherent with the finding of cardiomyocyte formation. And he also carried out a series of Western blots and immunoprecipitation to show that the interaction between each of these were critical with and without the presence of the blue box mutant. So in summary, at this point, the blue box domain to us seems necessary for cardiomyocyte differentiation. And that was the motif that was most critical. It's necessary for YAP cytoplasmic localization during the mesoendoderm transition. And NF2 blue box domain turns out to be really important for the complex formation that involves these 3 different proteins. At the cell adherence junction, whether YAP whether YAP is there or not there seems unrelated to phosphostatus of NF2 on that serine residue, and it seems unrelated to its close or open conformation. I'm putting here a figure where Mick also carried out proximity ligation assays to show the interactions between each pair of these proteins in complex. So the summary cartoon here showing that in the transition from pluripotent cells to mesoendoderm with the activation of Wnt signaling pathways, one key protein that our work has shown is important is the presence of NF2, because NF2 at the cell adherence junction is necessary and responsible for anchoring YAP, phospho-YAP, and keeping YAP phosphorylated at the cytoplasm, where if it didn't, it would be sitting in the nucleus. If it didn't exist, NF2, it would mislocalize YAP into the nucleus, and with YAP in the nucleus would suppress the expression of mesoendoderm genes. So where the work is going today is because YAP is so much in the biology and pipeline for cardiomyocyte therapeutics. We believe that now there is a new player that we can add to this space with NF2, especially specific to its blue box motif. So there is some projects going on in the lab developing this towards potential therapeutics for YAP regulation in cardiomyocyte biology and disease. I mentioned that Mick was the main person driving the lab, but there was a lot of support that came from other members in the lab. Thank you very much for listening to my presentation, and I welcome all listeners from Japan to come to visit us in Singapore if they have a chance. Thank you very much.
Yeah, thank you, Roger, and a great presentation. I have a question about, I have so many questions, and is okay? Please, Professor Komuro.
Okay, I come back. Actually, we have recently reported that TED1 is really important for cardiomyocyte differentiation. We found that the patient who has lamin mutation, that the mutated lamin trapped TED and leading to the downregulation of the transcription by TED. So I got a little bit confused that NF2 knockout, so dephosphorylated YAP goes into the nucleus and activates the transcription with TED, but it's not good for cardiomyocyte differentiation.
No, so it's in this transition from mesoendoderm, from pluripotent cell to mesoendoderm, you need to move YAP out of the nucleus so that it would allow mesoendoderm genes to get expressed. But I think you are pointing out the very intricate biology and pathways with YAP localization, because if we go further into the transitions to progenitor to cardiomyocyte, there's actually very intricate differences to where YAP should be in the cell. But what we find is the transition to mesoendoderm critically needs YAP to come out of the nucleus.
Okay, I got it. The story is similar to the Wnt story. More than 10 years ago, we reported that Wnt is really important for the differentiation from mesoderm to cardiac mesoderm, but it inhibits the differentiation from cardiac mesoderm to cardiomyocyte. So there are two phases in the Wnt. The probably YAP is the same, right?
Yeah, exactly. One is activating and one is inhibiting. You need, yes.
It depends on the timing of the differentiation. Yes. Thank you very much.
Yeah, I also think that this mechanism is context dependent. So, yeah, my question is about if you delete NF2, what cell types your cells differentiate into what cell types?
Yeah,
You in your story, yeah, you mentioned that in NF2 knockout cells sustained pluripotency.
yeah, no,
And this mechanism has been confirmed by transcriptional analysis.
no, not really. I think it becomes a very messy cell that is very hard to understand using the markers. It looks like epithelial, definitely non-mesoderm, non-endoderm type cells. It's not cardiomyocyte. No markers are coming up. There are some epithelial markers. There is some level of pluripotency still, but it's not entirely pluripotent also. The reason why we think the cardiac disease is not found in neurofibromatosis patients is because the mutation to cause cardiac disease needs to be in the blue box motif.
Mhm.
If your mutation is outside blue box motif and it's just giving loss of function to NF2, it probably is still fine for cardiac differentiation.
Oh, great. My another question is this story related with the phenotype of human patients with NF2 mutation?
Yeah, no. So far we couldn't find any patient with mutation in the blue box domain. So a patient with mutation outside blue box domain, they all have neurofibromatosis with neuronal tumors, but no heart involvement.
The interaction between NF2 and NF2 YAP and with beta-catenin, these interaction may be related with the human phenotype.
Yeah, it seems the interaction that is necessary for YAP localization to cytoplasm doesn't get disturbed by all the other mutations that we see so far. It's only disturbed by mutation in the blue box domain.
Mm.
It may be, we think, that the blue box and firm domain is needed for interaction with LATs, because some people also find LATs kinase in the cell adherence junction. And the LATs is required for phosphorylation of for YAP as well as maybe also MOT.
Christine, are there any questions? You have already known the story?
Yeah. Roger. Yeah, very interesting. So I'm following on actually, what do you think about the potential of your finding to do in vivo direct transdifferentiation, say cardiac fibroblast to cardiomyocyte? Yeah, how can that be? I think experimentally it might be possible. Yeah. What do you think?
Yeah, that's a good question. So I didn't think of the transdifferentiation model so much. I was more thinking about the regenerative medicine model because, someone like James Martin and other people are trying to use YAP signaling to induce cardiomyocyte proliferation. So they knock out Hippo and induce YAP for proliferation. So in that direction, we are thinking we may also contribute by some kind of NF2 regulatory level upstream and not involving Hippo, if this is so cardiomyocyte specific. On the note on your question about the transdifferentiation from fibroblasts, I didn't, to be honest, I didn't think about that too much yet, but I will have to think about that. I mean, that one is using the GHMT, GATA4, HAN2, and so on, right? To transdifferentiate. Already the efficiency is up to 30% in vivo if you believe Deepak's work. And I don't know if YAP has any contribution towards increasing that. I need to go and read the literature. I'm not familiar.
See, yeah, thank you. I think there's just a lot of exciting prospects. Yeah.
So thank you, Roger, for great presentation. So finally, I'd like Dr. Roger Fu for closing remarks.
Okay.
okay.
Thank you very much, Seitaro. Actually, you should invite Professor Komuro to do closing remarks. I'm really privileged to participate in APSC with your Japanese colleagues. It's been actually fantastic to know that APSC is so active among the Japanese community. Precision medicine, like Professor Komuro said in the early part of his introduction, is a really important new frontier for us in practicing cardiology. New drugs are going to come, new ways to stratify patients. We see already drug like mavacamten coming to the clinic for myosin therapy, directed therapy. So we're really excited in the new space forward. And so we saw so many things in our presentation, from atrial fibrillation to vascular smooth muscle cells, endothelial cells, to Seitaro's targets in different genes. So very excited and look forward to more interaction with the community. Thank you so much for inviting me and Christine. So do we close this meeting here, Seitaro? Thank you.
Yeah.
Thank you very much, Roger.
Thank you very much.
Thank you. Bye-bye.
Bye.
Bye-bye.
Have a nice day. See you.
Thank you.